on like every single multiple choice free response thing I've looked at, enzymes comes up all the time. So let's talk about, first of all, just the structure of enzymes. What kind of structure do they have? What macromolecule are they?
Enzymes amino
Proteins. Okay.
Well, yeah.
Remember, proteins have like that quaternary structure, so they have the amino acids, and then they either beta pleat or alpha helix, and then they come together to like the tertiary form, and then you're adding in a bunch of different ones for the quaternary form. So really complex structure. What does that complex structure mean for like heating it up? What happens to the bonds involved in that?
They catalyze one another.
Well, not catalyze one another, but they get cut off, right? So, like if this is the enzyme that we're talking about, and it's quaternary, so there's a couple different protein strains coming in here, and what's actually holding together this structure is just interactions between like each other. Okay, so like maybe there's hydrogen bonds that like are gluing these molecules together. And when you heat one of these bonds, like it's likely to break. Okay, so when the bond breaks and this molecule starts to fall apart, what do we call it? Heating causes denaturing. Yeah. So heating causes denaturing, which again just breaks these bonds. So once you break that bond, you kind of like make that enzyme not work. Okay? So what happens if the enzymes get too cold though? What happens to any molecule if it gets too cold?
It does. Like it stops moving.
It stops moving, yeah. So cooling decreases the collisions, the number of collisions. So just like, that's the reason why you stir stuff. Like if you're cooking and like you're stirring and you have to heat something up in order for it to like dissolve into the water. So for example, sugar and water. Like if you just pour sugar into water, all the sugar is gonna go to the bottom. It's not until you stir it up or you like heat it up till you like increase the energy enough that it like dissolves. Okay, so too much cooling decreases the number of collisions, which means the enzymes kind of work at an ideal temperature. So so enzyme activity is really low when the temperature is low, and then you kind of have an ideal temperature, and then the enzyme activity decreases when you get too hot too. So beyond here it's denaturing, and beyond here it's just too cold, so number of collisions is low. Okay, everybody good with that? Okay, remember that pH also does kind of the same thing. So most enzymes have an ideal pH. So this is enzyme activity here. So most enzymes have an ideal pH. If the pH is too high, you're going to kind of see the same thing, that it denatures. And again, that's by breaking the bonds that are holding this quaternary structure together. And if the pH is too low, you're going to see the same thing here too. Okay.
What did you say? EA is enzyme activity?
Yeah, enzyme activity. Okay, so the one thing that doesn't follow this, so I'll go ahead and say pH is maybe specific to the enzyme. So like certain enzymes work at better pHs than others. So like enzymes in your stomach are going to be able to function at a really low pH, because keep in mind your stomach has a really low pH, so those enzymes are almost like perfectly fit for that environment. So you might see that the peak is like at pH 2 and then decreases like that. And then enzymes that are working like in your spit or in your blood are going to work at slightly different pHs. The one chart that doesn't look like this for pH Anybody remember when you increase concentration of solute, or of substrate, I'm sorry. What is a substrate? Come on.
It's like the building block of the enzyme.
The substrate is the surface, or what's going on the enzyme.
Yeah, so what's binding with the enzyme? So if I draw an enzyme as like an E, here the substrate is going to be what binds. So remember, enzymes are really substrate specific, so like it has to be a perfect fit. And granted, like I'm kind of simplifying this here. Really, we're talking about the enzyme having a really complex structure, but still you end up with what's called an active site.
What unit is all this in again?
Macromolecules. Okay, so you end up with an active site that's really shape specific to the substrate. Okay, so the lab that we were messing with like filter paper and yeast. Do you remember that lab? And we increased the concentration of yeast that the filter paper was soaking in. Everybody can remember this?
Yeah.
And so then what we would expect to see is that if you increase the amount of substrate, so if this is number of substrate molecules that you have, and this is again enzyme activity, what you see is that like you can increase the substrate concentration, but eventually it's going to level out. Why is it going to level out? Like why do we see that does that way?
Because I mean, no matter, if it's the right pair, will go up. But if it's, I mean, eventually it's gonna level out because we just keep finding the same pairs.
So yeah, you're onto something there. So like even if you increase the number of substrates that you have, like you still can't speed up this enzyme. Like it's going as fast as it possibly can. And remember, like it's grabbing onto people and then ripping it in half, and then it just keeps kind of randomly with its eyes closed bumping around. And then when it runs into the next substrate, it rips it in half. So if there's a lot of substrates, like you're probably gonna have less time in between substrates, but like you can only go so fast. The enzyme can only work so hard. And like you can't possibly like make it work harder. So the concentration of substrate definitely like increases, and then once it gets to a certain point, levels out. And we saw that in that lab. I don't know if you guys remember it, but we saw it kind of increase, and then it stopped. I think there was one point that like didn't follow this line in our lab, at least for one class. That's how it did. Okay, any questions on enzymes? We okay with this? Okay, Jack, can you pause that?
Yeah.
The phone.
Oh.